It's 7 o'clock. Let's get going. Hi, I'm Vivian New. I'm one of the co-presidents for the Santa Clara Valley chapter of the California Native Plant Society, and I wanted to welcome you tonight to our talk, Bird Nests as a Botanical Time Capsules by Dr. Justen Whittall from Santa Clara University. And I would first like to acknowledge that the work done by the Santa Clara Valley chapter of CNPS lies in the homeland of the Amamutsan tribal band, the Tamian Nation, the Ramaya Tushuloni, and the Muwekma Ohlone, who still live and thrive in this area today. We hope to learn from them and support their work to restore traditional practices and to heal from historical trauma. If this is your first talk with us, we would love to know how you found out about us. So if you're willing, please share that information in chat. And this is streaming both on YouTube and on Zoom, and we are monitoring chat in both locations. So feel free to share any questions you have in chat, and we will take them either at the end or during Dr. Whittall's presentation, depending on the type of question. If you're not familiar with the California Native Plant Society, we are a nonprofit environmental organization that was founded in 1965. We have over 13,000 members in 36 chapters that are spread all over California and Baja California. So we are not just a state organization, we're actually international as well. Our chapter is the Santa Clara Valley chapter, which covers all of Santa Clara and southern San Mateo County. So we go up to about San Bruno Mountain. And our mission is to protect California's native plants, their natural habitats today and into the future. And we do that through science, education, stewardship, gardening, and advocacy. If you're not currently a member, we would love to have your support of our movement to conserve California's native plant diversity. And as a member, you would receive not just one, but two magazines, Artemisia, which has science-oriented articles, and Flora, which is general interest, has a lot of great gardening articles, a lot of beautiful pictures. And you'll also receive Blazing Star, which is our chapter's newsletter and tells you about upcoming events. You'll also get discounts at local participating nurseries. So it's very, very easy to join. Just go to cnps.org/join, or if you have a QR code reader, you can snap a picture of that QR code and that'll take you directly to the place to sign up online. We're a busy chapter. We have a lot of things going on. Most Saturdays, there are work days at 3 parks in San Jose, Lake Cunningham, Cataldi Park, and Kirk Park. Be sure to check on our meetup to see whether or not the workday is actually on for the particular Saturday that you're interested in. We also have a number of field trips coming up. We have a Manzanita walk in Mount Madonna County Park, and I realize I forgot to put the date, but it is in February. Check meetup and you'll see when it is. And ditto, there is also a Durka walk at Windy Hill on February 15th. We also have, which I forgot to put on the slide, and I'm sorry about this, Native Plant ID, which is on the fourth Tuesday of every month at the Peninsula Conservation Center in Palo Alto. And we also have a photo ID group, a photo sharing meeting on the third Friday, but don't quote me on that, of every month. So check on Meetup because that's where all that information is. And the information is right there on the slide. But you can also always go to cnps-scv.org to find out more information. If you're not currently on our chapter news mailing list, I strongly advise joining because then you'll get in your email once a week a message that tells you about all the upcoming activities. So you don't have to remember all the things I just told you because you'll get it in your email in a timely fashion. And it's very easy to do that. You just send an email to this long email address, but you can find that email address on our website at cnps-scv.org if you haven't copied it from the slide here. And now, before we get started on Justin's talk, I just wanted to encourage everybody to mute your microphones. If you have any questions or comments, please type them into chat at any time. It doesn't matter if you're on YouTube or on Zoom, we'll catch them. We will finish definitely by 9 o'clock, possibly earlier. But again, it depends really on the number of questions we receive. So please do share if you have any questions. I know Justin is quite willing to answer anything that comes up. And oh, I forgot to mention too that on YouTube it is being live streamed now, but that recording will be available for viewing immediately. So if you want to view it again right after we finish, you can do that on YouTube. But you can also just watch it later too. And so tonight's program, as I mentioned before, is Bird Nests as Botanical Time Capsules by Dr. Justen Whittall. Justin was actually one of the very first people we had give a talk when we started these Zoom talks back in 2020. He did that on jewel flowers because he also studies jewel flowers. But this talk, I think, is truly fascinating, not just for people who are interested in plants, but also in birds, because he has been studying bird nests and finding out all kinds of interesting things. And so, Justin, I'm going to turn it over to you, and I'm going to stop my share now. And it's all yours. Thank you, Justin.
Awesome. Thank you so much. Let me get my share going. Vivian and Priya for having me. Can you hear me okay?
Yes.
Perfect. And I wanted to thank you for inviting me back. And the last time I was on Zoom, I was outside in a patio trying to use the neighbor's Wi-Fi because we were all so new to it. This time we have it all figured out, and I hope things go smoothly. If at any point in time it's not, you can't hear me clearly, just let me know. I would like to talk to you tonight about time travel. Now, I've always been fascinated with time travel, and I think at some point everyone's thought about going back in time, and we could solve so many problems from the origins of the life on Earth to the first branches of when eukaryotes split from the bacteria. We could then see the end Permian 500 million years ago when 90% of life blinked out and how close we got to losing it all. We could, of course, catch the meteorite that landed off the coast of the Yucatan and see all of that go down to some more recent things and maybe more interesting to our own lineage, like when there were 6 species of our own genus living in portions of Europe and coexisting. Were they friends or foes? Time travel and the idea of getting insights into the past has always been fascinating to me, although I know our opportunities are often limited and just glimpses. They're often just snapshots in fossils or evidence from tree rings, for example. And what I'd like to talk to you tonight is a story that was inspired by a student who came to me asking for my help. And I was sure that Alex Rinkert, who was a graduate student at San Jose State at the time and my co-author on this particular talk, came to me and asked me if I was interested in time travel. Of course I was. And in particular, he said that he would like to recreate the plants that grew around the San Francisco Bay over 100 years ago. And I thought he was surely in the wrong place because I didn't know anything about that. And then he said he would like to study, he's a bird expert. Alex is writing the breeding bird atlas of Santa Cruz County right now. I'm almost finished with that. And so then I was sure he was in the wrong place when he suggested that we were going to use bird's nest, because I know even less about bird's nest than I do about the plants around the San Francisco Bay estuary. But when I put two and two together and realized, oh, what he's looking for is a place where we can maybe identify the plants that are in the bird's nest. When he layered on top of that, the ingredient that many birds' nests in museums like the Cal Academy and at UC Berkeley and across the country, it turns out, have bird nest collections that are over 100 years old with some specific geographic locations, suspiciously specific given the fact that they're over 100 years old. And now I'm interested and maybe even slightly qualified, as we in my lab study a lot of different types of plants and other questions in molecular ecology. And frequently we gain insights in addition to a lot of field work, which I have the most respect for, but we can gain some insights, especially about the past, by looking at their DNA. And we've extracted DNA from hummingbird pollinated columbines, which happens to be a subject close to my heart and something that I had the opportunity to talk to this very group about when I first returned to Santa Clara after having grown up here as a young professor at the Los Altos Public Library. And we've extracted DNA from rare mustards like the endangered wallflowers, as well as the Metcalf Canyon and most beautiful jewel flowers of southern Santa Clara County, which we are still working on preserving, and our restoration work there continues somewhat successfully. We extract DNA from the most endangered pines in the world. That's the Torrey pine from La Jolla and the Channel Islands. We've worked on DNA of things that aren't even plants, like some medicinal mushrooms like reishi, and helped with the herbal supplement community in that respect. From the insects that spin webs, the finest silk comes from amphibians, and I've helped a professor out, and we've extracted their DNA. And even most recently, I've been working with some students on the microbiomes of endurance athletes, because when I'm not out looking for plants, or sometimes when I am looking for plants, I end up on a long run, and they were curious about what grows in their stomachs. But all these things made me realize, oh, I do have some experience extracting DNA, but I've never tried to get DNA out of anything that was older than a couple of decades in from an herbarium specimen, for example. So this was going to be a unique challenge. Before I go into the details of how we did that, I'd like to give you a little justification. Why is this interesting? And some of this will surely be familiar to a lot of your audience, but let's make sure we have the setup proper here. 1 second. Oh, so today, that's Alex Rankard, by the way, in the top right corner there, and he's sampling a nest from the Cal Academy. Down below, you'll see Bear Island. That's off the east coast, east side of Highway 101 in Redwood City, looking northward at San Bruno Mountain, and I think that's the San Mateo Hayward Bridge in the distance, and you can see quite a bit of water inundating it. I'd like to tell you about the habitat as a very specific habitat on the margins of the San Francisco Bay, and how we're going to use birds' nests to maybe help inform restoration practitioners as to what could be planted there. I'll show you a little bit about how we got the DNA out and what works well and what doesn't work. We've learned a lot about that. And most interestingly, I think for me and hopefully your audience here, you guys, is the plants that we found. And in some cases, the plants that we didn't find. So here we go. Of course, wetlands around the San Francisco Bay Area are extremely endangered. At one point in time, predicted in, say, the 1800s, wetlands occupied substantial areas in the South Bay, from Redwood City through Alviso to Milpitas, in the North Bay, San Pablo Bay, on the way to Napa and Sonoma up here, for example, and even Sassoon Bay, including Grizzly Island. But presently, in green here, are the remnants of those extant wetlands, and you'll see very few spots remain. There are little tiny slivers on the edges and little tiny refugia, but not much left in terms of the wetlands. The wetlands have been converted from everything from salt ponds, which were established in the early 1900s and really became expansive in the mid 1900s. But we now have quite a bit of pavement and development all around the margins of the San Francisco Bay. But here you can see an example of a little swath here off in the South Bay of what wetlands may have looked like. Let me show you a couple of cartoons that describe a very unique habitat that is on the margins of these estuaries, not the actual wetland itself, which we know is a biodiversity hotspot, very rich in organisms as well as a very important ecosystem for ecosystem functioning. But let me tell you about the transitional habitat, because the transitional habitat is particularly unique. And the transitional habitat is the space between the highest of the high tides, which barely get into the transitional habitat, and the high king tides, which only occur a couple times a year, and they will inundate the transitional habitat. So that space between the high tide line and the king tide line is called the transitional habitat. Below that, you have wetlands, here in light blue, for example. And above that, you have the uplands where you're start getting with what we thought was more woody, shrubby, and occasional tree-like habitats. The transitional habitat today is often extremely compressed because of the levee system, such that you go over this real subtle elevational change over a very short geographical distance. And that therefore compresses our transitional habitat into a small place. And when it's there, it's rarely intact. It's almost always filled with invasive plants. Here's a picture of what it might look like at Bear Island again, and you'll see the line for the high tide line with some extremely salt adapted plants down at the lower line. Between the two orange dotted lines is the transitional habitat. Occasional woody plants appear there, most of these introduced, and then the upland levee top. And this was expected to expand quite broad in some places, especially in the South Bay, to maybe a couple of kilometers, and is now often only a couple meters wide. For example, here's a estimate from a 1926 paper. This is a secondhand paper we're going to see a couple times by Cooper, who took some storytelling from a couple of long-term residents of Alvisa and put those together with some historical records to try to estimate as much as Cooper could from the habitat of the transitional habitat. And you'll see in orange is the transitional habitat. And in some places it's relatively small. In blue is Highway 101 presently, of course, not there in the late 1800s when he was trying to make these estimates of. But you'll see how wide it was here around Mountain View, for example. This would be the San Francisco Bay. This would be the high tide flood zone. And then the king tides would have come all the way out, way past Highway 101. The accuracy there, I'm not going to put any of those things on a map, but I just want to emphasize that these things were expansive and therefore an entire set of organisms that were unique to these habitats would have existed. They provided really important biological functioning. For example, in these areas, there's rich biodiversity, deep soils, lots of different species. As you go from one habitat to another, that's called a cline transition. And we now know that they will play an important role in our currently and near future. With sea level rise, we will see these transitional habitats or places where water can be absorbed, picked up by plants, evaporated, such that we can prevent flooding. Here you can see, for example, with sea level rise shown in the dark blue compared to the top there, normally plants would just move up. The transitional habitat also provides refugia or a hiding place for a lot of unique organisms. And although this is a talk primarily about plants, we know that plants interact very intimately with things with backbones that get a lot more attention and sometimes even more protection. For example, Ridgway's rail with its distinctive black and white bars on the haunches here of the leg. It has particularly long toes to get around in the muddy flats and somewhat slender legs so that it doesn't fall through the mud. Ridgway's rail has a unique salt gland that allows it to drink salt water, and it uses the transitional habitat as its refugia during those high tide events. The salt marsh harvest mouse is a listed species. It comes in at 6 inches long, but half of that of it is its tail, weighing only half of an ounce for the entire thing. We're talking about a relatively small mouse here, pictured amongst some particularly red pickleweed. And the salt marsh harvest mouse uses the transitional habitat as a refugia. Let me give you a little visual here. Imagine you're that 6-inch harvest mouse and you're enjoying your life amongst the pickleweed, either over here or over here. And during a high tide, no problem. You come to the edge and maybe over here, you find a little dry spot and wait out a couple hours and you'll be back. But if it happens to be a king tide, your habitat will now look like this. And as a 6-inch mouse, you may have a challenge getting from what was your refugia across that region, wet region, in order to get to the transitional habitat. And so the more expansive these transitional habitats can be, the more habitat we can provide for endangered animals like salt marsh harvest mouse and things like the ridgeways rail. With king tides, you have less refugia, therefore more chances of predation on endangered species and maybe decreases in the population size. So, not surprisingly, people are interested in restoring the transitional habitat, not only for its own sake, but also as habitat for some vertebrates. Right here is a nice map of the San Francisco Bay Estuary, and you can see in gold are regions that are predicted from a 2019 study that could be candidates for restoration of the transitional habitat, which is encouraging. Lots of possible space that would potentially be useful for transitional habitat. Also, lots of stakeholders interested either by choice or by force in helping out with the restoration of these transitional habitats. Unfortunately, we have very little specific knowledge about the plant species that live in the transitional habitat, which is a surprise to me. I thought, well, we'll just go to the herbarium and we'll find some candidates and look for the oldest specimens and sort them by age, and we'll quickly get a list of things that were there before the arrival of the Spanish and all of the joys they brought from Western Europe. Unfortunately, the details on a lot of those herbarium records are super vague. They might say San Francisco Bay. They might just say Northern California. Some of them just say California, or sometimes no, Northern Mexico. So very little details about what grows in the transitional habitat as to be distinct from the uplands and the actual wetlands itself. At the same time, there were not very many botanical surveys that would allow us to sort of record in the literature, the scientific literature at the time. We don't have very many reference sites remaining. That would be a very specific location. Parts of Grizzly Island, parts of Bear Island can be helpful. The lack of herbarium specimens with the habitat details, without books about the habitat from those times. And lastly, this Cooper paper from 1926 is a very beautiful and eloquently penned description of what he thought the habitat may have been like from secondhand storytelling from some Alviso residents, but it's not very scientific. And I'm going to show you the list here, and you'll see some of the great things about it, but some of the limitations. For example, Cooper's paper in ecology suggested the following species occupied the transitional habitat. There were 10 of them in the list, which we hope underestimates the diversity of the transitional habitat, maybe by an order of magnitude. And it doesn't really tell you much about the entire estuary. We know the San Francisco Bay is quite complex and it has multiple players in the south versus the north versus the east. And some of you may recognize a couple of these plants by their scientific names. In fact, they are particularly enriched in the sunflowers, and I think all but two or three of them, in fact, are in the sunflower family. They also are things that you would tend to see more likely in the summertime than in other times of the year. And I feel like if we just planted these things in the transitional habitat, it would be extremely colorful during the late spring and early summer, yet it would probably not represent the diversity. We expect this transitional habitat to be rich in grasses, for example. And I wanted to point out a couple of species that I didn't expect to see, like this Rosa californica, the California wild rose. And there's also a nice shrubby Baccharis glutinosa up here. We'll revisit those again later. Okay, so a little bit of information, but definitely not enough for restoration practitioners to recreate this transitional habitat, or at least create something that we think is functionally comparable to what was there before. That's where Alex and his bird skills combined with adept field notes and passion for the San Francisco Bay Estuary came to me and said, I know that there are museums that contain not just a few, but hundreds of birds' nests. They may be botanical time capsules for us because I know some of them are quite old. Pictured here is a song sparrow nest collected in 1915 by Joseph Grinnell. You may know Grinnell's name if you've spent time looking at animals. I consider him the Jepson of California zoology, although I'm sure many zoologists would be offended by that, but I think that's the biggest honor that I could give him. Jepson being one of the grandfathers of California flora. Grinnell was the one of the early bird, particularly, and also small mammal scientist, zoologist of California, and the first director of the UC Berkeley Museum of Vertebrate Zoology. Regardless, he was the collector here in 1915, now a 110-year-old nest. This is a photo that Alex took just a few years ago, and it seemed like it may not have seen the light of day for probably about 100 of those 110 years. There were no signs that anyone had checked it out or had been on the list to examine it or had annotated anything on the label. It it turns out there's a lot of old nests. So these are the collection eras from the 1870s in groups of 10 years, all the way through the 1930s, which by this point in time, the Spanish invasion, we'll call it, had already really taken hold. So we limited ourselves to the 1930s as the latest date and the 1870s as the earliest date because there were no more before that. And you'll see these are the number of nest specimens that we think we can track down to what seems like very likely transitional habitat. There are a couple hundred. Now, keep in mind that these nests are built, number one, from dead plants, and then they are probably abandoned and finally inundated multiple times by king tides before a collector has found them. And then they went to a museum where they sat for potentially up to or over 100 years. So we might be able to say some pieces are grass, other pieces are not grass. We might even be able to say they're not woody, so they might be a forb. But as we know from the hyperdiversity in California, and especially around the margins of the San Francisco Bay, that that is definitely not enough detail to inform restoration practitioners. And to be candid, this is what we're looking at here. Maybe the inflorescence of a grass fragment, potentially something segmented here, like the joints of a salicornia, that would be like a pickleweed, maybe another piece of grass fragment, but we couldn't get very far. We also know that these nests are precious. They're precious historical collections that capture a moment in time, and we don't want to disturb the entire nest, but we know that with DNA markers, we only need a very small amount. These are irreplaceable historical collections. So we're up to pursuing the extraction of DNA from the nests. The goal here then is to see if we can't take a couple small fragments of the nest, extract the DNA from it, and using a technique called the polymerase chain reaction, we will make copies, thousands or hopefully a couple million copies of that small fragment that we're interested in that differentiates closely related species. Maybe we'll find a tarweed in there or a tar plant, and that could be used to help inform what grows in the transitional habitat. I'll tell you a little bit about a couple plants that we discovered in our pilot study that's published, and then I'd like to finish with some preliminary analysis with a new technology that has grown the number of plants identified by an order of magnitude. I want to tell you a little bit about what works and what didn't work. But first, I need to tell you about a little bit about the birds. So you can't just use any bird nest found in the transitional habitat because a lot of birds, they fly far and wide to find their pieces of their nests, and then they might end up choosing the transitional habitat for their final nesting place, but we can't be sure that that's their preferred nest making materials. Alex, with his brilliant bird experience, realized that the savannah and the song sparrow are famously patriotic to the transitional habitat. That is, they arrive there, they seek for plants for their nest within that area, and then they make their nest there. And unfortunately, that's led to substantial declines, particularly in those birds. Alex did a nice survey measuring the distance from the nest that these birds were getting nest materials from. You can imagine the chances of finding a nest is rare. The chances of finding a bird making a nest is, I think, even rarer. And then the chances of being able to follow that bird from the time that they leave their nest, they get a piece of material and come back to their nest. I would only trust a bird expert, definitely not myself. And Alex has that those wicked skills. So he found that these birds tended to go 17 meters more or less, give or take a couple meters. They tended to be going sort of along the transitional habitat rather than up to the uplands and down into the wetland, which was encouraging. Some of them went as far as 20 meters, but most of them were within about 10 meters. This is really encouraging, although a small sample size, that we're going to be finding nests of unique birds that tend to fill their nests with plants from the right habitat. So here's our preliminary study. This was a proof of concepts to see if we could get it to work. Can we go back in time? We have two historical nests from 1915. You'll see those in purple here. One over in the Oakland area, I think, and then the other one up here in the Grizzly Island area. The two contemporary nests are from the South Bay, the Bear Island area, and down by Alviso. The contemporary nests are from 2003 and 2017. The historical nests are both from the same year in 2015. One of them is that Grinnell nest I showed you. Okay, let's have a look. In the museums, when we go look at these nests, it becomes very obvious that these nests are architectural dreams. They have all the things that you would expect in something that requires some structure. So the outer portion is often filled with somewhat woody or more heavily fibrous materials. And then the inner structure, often much finer materials, for example, a lot of grasses in the center, and maybe more herbs and potentially an occasional twig with some wood in it in the outside. And the outside, you'll often also find more coarse materials that could include some reeds or some large leaves of forbs. We systematically studied them, calling the outside the structure and the inside the lining. We took 20 samples per nest. It's really important that as we take these samples out, they are contiguous pieces of plants. So we don't want to have mixed DNA pools. So we tried to have just one piece of plant. Turns out that's a great start, but there is DNA just about everywhere all the time, whether you see it or not, whether it's a contiguous piece of grass, there's DNA from just about everything in the environment on that. And if your pinky is about 1 centimeter wide on average, so we're talking about relatively small fragments to try to keep the nest intact as a historical specimen. And these things, we went through every one of them, and the study grew exponentially by the end. And we've seen now thousands of these little fragments, and we can quickly get them into probably grass or not grass, but not much further than that. Okay, very briefly, this is the type of work that can be done in my lab at Santa Clara University. We can take these little fragments, we can extract their DNA. We use some kits. We use a lot of controls in place. For example, in addition to extracting the DNA from the plant material, we always have a water control such that we know there's no DNA is intended to go in there. Because the next step is an amplification process. We're going to try to make millions of copies out of just a few highly fragmented copies, and therefore any contamination could quickly become what looks to be a sample. So we also do a water control in the polymerase chain reaction. That's a negative control to make sure that it never produces any PCR products. We're using a particular region called the internal transcribed spacer. It's not too important, the details of it. It is not a portion of the genome that makes, flower color or flower or plants drought resistant or salt resistant. It's just a region that spaces out two very important chunks that make the ribosome. The spacer part is important because it means it can accumulate mutations over time at a relatively constant rate without compromising the organism. So they're basically like a molecular clock ticking away. And it works really well to differentiate close relatives. So we should be able to, if we can get something to amplify and sequence, use it kind of as a barcode, the same way you would take a bag of chips and put it across the zebra stripe across the instrument in the grocery store. We'll use the DNA sequence as the barcode to try to identify the species. We'll run a quick agarose gel and see if we can find a single band. The single band tells us that we have probably one thing amplified. And from that, we can send it to sequencing. We use a company in Mountain View, which ironically is off 101, not too far from where I expect the transitional habitat to have once existed. Okay, and then we do have to do quite a bit of computational biology. These DNA sequences are only as good as our reference panel. So we need a reference panel. We call it GenBank. GenBank is owned by the National Center for Biotechnology, part of the NIH, and it's in Maryland, and it has a repository for every DNA sequence that has ever been published, and even a lot that are unpublished. We found that we made a list of all the things that we could think would potentially be in the transitional habitat. We talked to people at SFBBO, we talked to people working for Mid-Peninsula, and a lot of people doing restoration around the margins of the San Francisco Bay. And we came up with a list with about 90 to 100 species that we thought could potentially be there. And it looked like 9 out of 10 of them had a unique record in GenBank, that is, for that particular species. And if it wasn't that particular species, there was always a close relative in GenBank. Often the close relative doesn't live in San Francisco, so you can then leverage some sort of mystery solving skills and natural history to determine which species it is. We can access GenBank using a program called BLAST, and BLAST is a super powerful algorithm for identifying matches to a database of billions of DNA sequences. And we download all the most similar matches. We report what the top match is, but we also download them and we do some studies of genetic identity and even build some phylogenetic trees, which I'll walk you through because they tell quite a bit about the past. This is part of my time travel, is to be able to look at these trees and go back in time and work our way backwards. So at the end of the day, we're going to see some BLAST results, some genetic identity, and some phylogenetic trees. And here's our preliminary results. We found about 13 samples that made a single strong band out of 60, which was pretty low, less than 25%, closer to 20% success rate. And these are some of the plants that we found. There were 6 plants, there were 6 green algae, and even a ciliate, which is a protist. I'll tell you more about that in a moment. But you might recognize a couple of these plants, for example. The California rose came from the 100-year-old bird nest from Joseph Grinnell. It was also on the 1926 paper as a potential member of that habitat. Although I think a lot of restoration practitioners and myself were pretty surprised to have found roses in that habitat. Let me tell you a couple anecdotes about a couple of these plants. We've got a small Laemus microstachys and some Elamus triticioides here. And we've got cardamine and this pickleweed. Pickleweed, not surprising to have found DNA in that general area. Whether or not the bird was actually using pickleweed, we're pretty sure they were. I'll take each one of these individually and I think each one tells us a different story. So the 100-year-old rose from transitional habitat The ITS sequence, most likely Rosa californica, but we put that in parentheses because it required a little bit of natural history that I'd like to share with you, using some tools that I think many of you probably have used on the web regularly. It was also in the Cooper paper, which is encouraging that we were able to recapitulate a story from 1926, 99 years ago, which was actually a story that was 40 years before him. So here's a phylogenetic tree. Let me just orient you really quickly. When we take this Rosa californica sequence, send it to GenBank using BLAST, we can then pull down 100 of the most closely related sequences, and we build a large tree that looks like this. But if we zoom into this light blue box, we can see what is time on the x-axis, the horizontal here, and these are the sequences at the present, and you'll see some different roses from across the country, it turns out. And right here is Rosa californica, and there's our nest sample. The numbers here indicate the what's called a bootstrap value, and all we need to know for now, I'm glad to explain the details of how it's done, is that numbers of at or above 70% are usually considered a significant match. And this one was just below that, so we were a little concerned. It turns out that this sequence of Rosa californica is very similar, in fact, the same as another rose, which is Rosa nutkana. But having grown up around here and spent a lot of time in the woods, I knew I didn't see Rosa nutkana very often around the San Francisco Bay. I tended to see it on the coast, the northern coast of California, and actually quite common in the Pacific Northwest. Whereas here in California, particularly around the San Francisco Bay, Rosa californica is one of the most common shrubby roses. These little blue points show observations and herbarium specimens. There is like 4 Rosa nutkana in the San Francisco Bay, according to Cal Flora, and there is like 120 plus. I went on iNaturalist earlier today and I found 10 Rosa nutkanas and something like 2,000 Rosa californicas in the same general vicinity, meaning that there is probably a 200 to 1 chance that it was Rosa californica and not Rosa nutkana. Of course, the times have changed and we don't know for sure, but that's my suggestion. Here's what it actually looked like, which this was a perfect scenario. This is a 110-year-old nest, and there are the leaflets of the rose, which have this serrated edge, somewhat smooth margins at the base of the leaflet. Some of them were even still green. Of course, that just meant that they were preserved relatively rapidly after the plant was removed, which is kind of a perfect storm when it comes to old nests. You probably also recognize some other fragments back here, potentially a reed, for example, as well as some other grasses, which we'll get to in the later portion of the study. But that was the only thing that worked in the old nest on our first try. Let's take a quick change gears to thinking about the contemporary nest. Now, these are only about 10 or 15 years old, and we had much more success. We had 5 plants come from those nests. And let me tell you a little bit about Festuca microstachys. I think it also goes as Elaemus microstachys now. No, it's Festuca microstachys. It's definitely small fescue. It's a small, slender grass. And it seems like the perfect thing that you might find in the central portion, the lining of the nest, where the actual eggs are being held. The ITS clearly identified it as Festuca microstachys. It was not in the Cooper list, which had no grasses in it. And here's that phylogenetic analysis from our BLAST results. And again, just to orient yourself, these are 100 festucas. There are a lot of festucas. And I'll zoom into this little region where our nest sample landed, and there's Festuca microstachys, and the bootstrap value now significantly above 70. In fact, 100 is the best you can get, meaning we're highly confident that that sample was Festuca microstachys. Now, the exciting thing about this is, number one, it added it to the list, but number two, it actually came from a nest collected at Bear Island, which had been restored in 1917, and then the nest was found shortly thereafter. Before around 2017. In 2015, it was under restoration, and it looked like this. And we know that there was no Festuca microstachys known for the site from botanical surveys in the 2010 to 2015s. Festuca microstachys was on the list of those 100 or so plants that could be in the transitional habitat, so it was used in the restoration. And we found it in a rare bird's nest that's supposed to only use the transitional habitat. So I think that not only are we restoring the plants of the transitional habitat, but we're starting to see signs of having restored the interactions between the plants and the animals that use those plants, in particular, this rare bird. All right, Festuca microstachys, the anecdote on restoration. Here are some more results that we got. This is the Lamus or Elamus group. There are a lot of species. They're all really closely related and a little difficult to differentiate which species we have, but we definitely have ITS sequence of a Lamus Elamus. Those are not on the Cooper list. When we download the sequences, our nest samples come into two different groupings. They both seem to be associated with samples that look like Elamus triticoides are in the neighborhood. There's no strong bootstrap above 70%. This one's at 65. This one's over here at 65. So we can't be 100% confident, but with a little bit of natural history, we discovered that of all these other possible Elamus species, the only one that seemed like it could be in the San Francisco Bay margins was Lamus triticoides or Elamus triticoides. This represents a pretty exciting finding and yet another grass, which was what we were hoping to see, more grasses than originally suggested. But just to give you a sense of what these look like, these are the actual photos of what we extracted DNA from. This one is a small piece of culm, that's like the stem of a grass. And this one is a quite a bit larger piece, but we used it because it's contiguous, and you'll see the leaf here sheathing off here. I'm sure if you looked carefully, you could see those auricles that help identify it as the genus Elamus. But not everything was a plant that was native. So it's unclear the native status of Cardamine hirsuta in my mind, but I'm all ears if anybody has any insights. It's definitely not in the Cooper study, but it was 100% identity, and we have several samples from GenBank of Cardamine hirsuta, and this one lands right in the middle of them with a really strong bootstrap support. And then afterwards, we looked more carefully at the nest, and sure enough, you can see the little saliques. I don't know if you know this plant, but it comes out early in the spring. It's one of our firsties, grows all around disturbed places, and as you sort of brush along it with your feet or your hands, you can get those saliques to burst open and release their seeds. And looking more carefully, I said, "Oh, that's what that is. Those are long, skinny mustard pods called saliques." And not surprisingly, and sort of a positive control here, was the DNA from Salicornia pacifica or pickleweed. It's not on the Cooper list because it's more of a wetland than it is a transitional habitat plant. Super strong percent identity and bootstrap values are above 70. The exact species of Salicornia was a little challenging. Virginica, pacifica, or perennis. It turns out reading the literature about pickleweeds is probably about as tasty as the leaves themselves. It was described as a taxonomic nightmare come true. These things, I think, move widely with being so close to the water. There's northern and southern variants of Pacifica. This might be the northern variant. There weren't any records in the San Francisco Bay of some of these other species, so I'm saying it's most likely Salicornia perennis. We found 13 things that made PCR products. 6 of them were plants, which I've just walked you through most of them. But we also found some algae and a ciliate, which I thought, oh, we must have contamination in our water, and we got some green algae in the laboratory. And I don't know where the ciliate came from, but as we looked carefully at the sequences, we did see some patterns. The quality of the sequence was much lower. That is, it was very difficult to interpret these sequences than the plant samples. And all of these things, when we finally got names for them, were actually saltwater adapted algae and a saltwater adapted cilia. That's a little protestin with little tiny cilia that helps it move around in shallow, salty waters. We suggest that these were either incidentally deposited with the tides, as the nests were probably inundated multiple times before collected. Things like Ulva lobata there is one of the chlorophytes, for example. And those are some other green algae that we happen to identify. All of those sequences were different from each other, and they were all known from saltwater habitats. I doubt the birds were using them intentionally. They're most likely incidental. And there's the little ciliate stoma. It's in the Ciliophora, and its DNA is probably floating around. And whether or not it was intentionally or accidentally became part of the nest, or maybe it's just its DNA was washed up into the nest. I can't be sure. But we do know it was part of that habitat. And these are these little snapshots that I'd like to add to what we understand about the transitional habitat. Here's a couple little details in case anyone's excited. As far as we can tell in the literature, in the scientific literature, and the scientific community, we are the first to extract DNA from bird nests, particularly old bird nests, particularly old bird nests that are trying to be used for restoration. If you start with more mass, that is about 25 milligrams of tissue instead of 10, you have a much higher chance of a successful PCR. The white ones are the successful PCR, and the black ones were the failures. Again, there's about 3 times more of these than there are of these, and you'll see some error bars there. These are significantly different, meaning more tissue, more starting tissue, better chance of PCR working. We looked at the DNA concentration and we found that, not surprisingly, when you start with more tissue, you get higher DNA concentrations, more like 15 nanograms per microliter. That's how you measure concentration in DNA most of the time, instead of something like 7 or 8 nanograms per microliter. So more mass, more concentration. We thought maybe if we could measure the purity, that is, sometimes there are contaminants in the DNA extraction that can poison the PCR reaction. But turns out we measure the wavelengths at 260 nanometers and 280 nanometers. Those are in the ultraviolet, and that ratio of 1.8 is pure DNA, somewhere right around here. And both successful PCRs and not successful PCRs had not significantly different DNA purities, and they're both actually very pure, partly because we're using these DNA extraction kits that are famous for extracting pure DNA. So more mass, higher concentrations, don't worry about the purity. But I'd like to tell you now what we've done from this. So this is published in the PLOS One. It became Alex's master's degree at San Jose State. Go Spartans. And now we decided to move on from 4 nests, remember 2 ancient nests and 2 contemporary nests, to 20 nests. We're now using the same 2 species of birds, but these are 20 nests where we are highly confident that they, according to the records, came from the transitional habitat of the San Francisco Bay. They're mostly at or older than 100 years. 3/4 of them are from the song sparrow in the South Bay, and 1/4 of them are from savannah sparrows. And the nests came from a diversity of places requiring quite a bit of fun travel, locally here at the Cal Academy and the UC Berkeley Museum of Vertebrate Zoology, all the way down to Camarillo, where there is the world's largest collection of birds' nests, and the American Museum of Natural History and the Comparative Museum of Zoology at Harvard. Western Foundation for Vertebrate Zoology is in Camarillo, California, down by Santa Barbara. Okay, so now we have more nests and we have a new technology. We're going to use something called next-gen sequencing, which promises to overcome some of the limitations of the first approach. So we're still going to extract DNA the way we did it before. But previously, if we had multiple DNA fragments in our DNA extraction, our method probably would have failed. Using next-gen sequencing, we can sequence individual fragments at a time, even if they're in the tube together. For example, if there's a pickleweed and a small fescue DNA fragment in there, they would have failed in the first round, but using next-gen sequencing, we can overcome that. And finally, it's extremely sensitive to really small amounts of highly degraded DNA, which is exactly what we likely have. These are the technologies that promise your own genome for less than $100. We start with the genomic DNA. Ironically, it's already in small fragments. We still have to cut it down to be exactly the right size so we can add some known DNA sequences to it. Very briefly here, but somewhat interesting from a tech perspective, from a biotech perspective. We are going to then take these pieces of our DNA and we are going to attach them to a slide. That's like a microscope slide, very similar to what you would have used if you put some pond water and wanted to look under a microscope with a little cover slip. It's almost exactly like a microscope slide, except it has a lawn of these red and blue primers where our DNA will bind to through hydrogen bonding between the A's, T's, G's, and C's, and then under certain conditions undergo what's called bridge PCR, where it's actually doing amplification in little tiny colonies on there. And then using a fine microscope, it's going to take pictures every time it adds another base, an A, a T, a G, or a C. And it looks something like this. This is called sequencing by synthesis. And eventually the photo of the slide looks like this. And each one of these little glowing dots is either an A, T, or G, or C. There should be 4 colors in there. And we then grow that one base at a time. And next thing you know, we have our fragment of our region, and we can use the same bioinformatics tools we used previously to identify the plants. Much more sensitive and can deal with heterogeneous PCR products. Higher success rate, more plants, more information about the past. Let's go. This is what we found. Our next-gen sequencing approach, preliminary results, first divided into growth form. So we have 72 plants instead of just 6. We've grown it by over 10 times. This is the percent of those 72 plants in savannah sparrow nests or song sparrow nests on the x-axis. And I've just split it up quickly into life forms because I was really surprised. I thought we were going to get primarily grasses and herbs, but it turns out shrubs and trees occupy between 20 and 25% of the nest materials. Of course, grasses and herbs explain about 75 to 80% of the nest materials. Let me tell you a little bit about native versus non-native plants over the years. So this is the time machine. We have nests as far back as 1875 in groups of 5 years here through the 1920s, with a couple of missing years where we don't have nests for. And this is the percent native versus non-native. And we have 100% of the plants in 1875 native, and you'll see that quickly drops to around 80, and then it kind of fluctuates between, say, high 80s and the high 70s all the way through the 1920s. Of course, the contemporary nest, the percent nativeness was closer to about 50% and or below. So this is very exciting. I think that we're actually getting a good glimpse of what's happening because we are seeing, 4 out of every 5 plant out of this nest was native, even though, of course, the Spanish had already been there for a couple hundred years, even at my institution, having built a mission there and brought all their accoutrement with them. Let's have a quick look at some of the plants that we found. Not surprisingly, there's pickleweed and some salt-loving plants like saltgrass, Disticus spicata. We find Spartina, Jaumia, Elymus triticoides, one we saw before, even California sagebrush, a shrub probably providing some of the outer architecture. Several of these were expected, but now we actually have evidence that they belong in the transitional habitat. We did find some non-natives, and I think some of them remind us that these things have been around for a long time. For example, one of the most common non-natives is Medicago polymorpha. You recognize the curly spine-tipped fruits there in that little legume. Lots of introduced grasses in the nest, like Poa annua and Festuca bromoides, which have been in California since those time periods. We saw Cranamina hirsuta before, but another mustard is a wild radish, which has been around for some time. A couple of uncertain sedges and linum, probably brought here for its fibrous uses. That's all very visual. I love to show plants, particularly when that's the subject. But I want to show you a graph that summarizes it all, and it's going to be big. There's going to be a lot of small font, just be warned. But before we go there, let me first explain what else we found in our results. Remember I mentioned how sensitive it was, highly sensitive? I'm not suggesting that these birds are making their nests out of bagels, but sesame seeds turned out to be one of the highest frequency hits that we had in the top 10 for plants. And it could have come from a lot of places, as could all of these things, which were found in our PCR products. They could have been as simple as the lunch that either my students had or the people who did the sequencing had. However, we always had these water controls and we never picked up any sesame seeds or almonds or oranges or cantaloupe in any of those negative controls. So I don't think it's something that we've done. However, there were people living around the edges of the San Francisco Bay and they were eating food. And some of these foods they were probably also not just eating, but growing and could have easily become part of the environmental DNA of the area. Um, we tried to put enough controls into place to exclude recent contamination, but we also don't know what's happened in the museum over the last 110 years. So um, with that caveat that there are some things in there that I know are not part of the native transitional habitat. Here's our list of 72 species of plants. How frequently they were found is on the x-axis. I can explain that axis in more detail if you're interested. It has to do with how frequently the sequences were read. The gray bars are the savannah sparrow and the black bars are the song sparrow, and they kind of fluctuate back and forth. And they're currently in order from the most common to the least common. And you'll see Salicornia, that's the pickleweed at the top, and Anemones, a grass, Triticioides near the top as well. There's a Medicago. And let me show you some of our old friends. There's the California rose, was actually found in multiple nests in both Savannah and song sparrow nests. That was exciting to see. It wasn't just a one-off from the Grinnell nest, that one sample. Some other really interesting, but maybe rarer nest materials that I've gotten excited about is a senecio. I can't tell what species it is. It might be triangularis, it might be hydrophilus, but that would be an exciting find for me to put that onto the restoration practitioners list. And a solidago that I also can't be sure of which species. It could be spathulata, but we need to do a little bit more sequencing of reference material to be sure what species it is. And that would also, I think, be a new perspective to some restoration practitioners. I was excited to see a lot of woody things in the list. So here's an unidentified pine. Now keep in mind that these are from DNA. So it just means that there was pine DNA. It doesn't mean they were necessarily using a needle, although we had a lot of materials that look like they could have been pine needles. But the difference between a 100-year-old pine needle and a 100-year-old piece of grass is subtle. One of them has joints in the culm, and the other one doesn't. But the birds don't always pick the pieces that are the most distinctive. We have some willows and oak DNA. We found redwood DNA, which we don't think is part of the transitional habitat. We don't recommend planting redwoods in the transitional habitat, but it does give us a glimpse of what DNA was floating around the San Francisco Bay over 100 years ago. Even eucalyptus, which I'm about to tell you about some plants that are noticeably absent, but eucalyptus was known to have been brought to California in the 1850s and spread widely in the 1880s, 1870s, and 80s, where they started planting large patches of eucalyptus. Some of those, I think, around Berkeley might be the oldest in the state. More woody things, but these more in the shrub life form. You'll see that California sagebrush at the top. You surely recognize the Ceanothus thurcifolius. And we found Baccharis, which was on the Cooper list from 1926. This is Baccharis gluten. Oh, we have two Baccharis. Glutinosa was a little bit more common than Pilolaris, but both found. And I think the glutinosa especially is a very good candidate for the transitional habitat. And not surprisingly, some salt-loving plants in the genus Atriplex that you'd expect from the margins of the San Francisco Bay wetlands. I wanted to show you some more plants because that really gets me excited and brings these results to life. a couple things that I was missing. I thought I took many walks and long runs around the edges of the San Francisco Bay. We're out there doing field work, and I know that these consume very huge amounts of acreage around the edges of the bay. For example, wild oats, which a colleague of mine says is probably the most common grass in the world. It's probably has the most number of individuals spread throughout the world, is Avena fatua, was not collected in California in their herbarium until 1950. It's a relatively recent arrival. You remember our nests stop around the 1930s. It wasn't in the bay until the 1970s, and it doesn't look like it was in any of our results. Nor did we have any Phragmites. Phragmites australis is a massive pest of the wetland ecosystem. It causes a lot of ecological disasters. And we know from a colleague of mine, Virginia Matzik here at Santa Clara, that it's probably only about 30 or 40 years having arrived in the estuary. And I wanted to mention Cutula coronifolia. That's brass buttons. It's a cute little plant, and I really wish it was native. It's actually being planted by people restoring portions of the wetlands for duck habitat, which was exciting that people care about ducks enough to plant plants for them. It was a little disappointing to know that they're planting non-native plants, and it definitely was not here 100 years ago. All right, a couple things that we're missing. So let me just give a quick summary, and then I can answer as many questions as you want. Can we use nests to go back in time and find out glimpses of the past using their DNA? For sure. Multiple iterations of this. We've done it with the traditional method and some next-gen sequencing. We can get nests as old as 110 years now, and some of them are visually completely unidentifiable. We know that if you use more material and higher concentration, you have a better chance of it working. And let me tell you briefly about some of the things that I for sure don't know. Is we are not providing an exhaustive list of everything that was in the transitional habitat. We're only getting things that were used by these particular birds. So again, it's a snapshot. We also are only picking things that would be likely to be used during nesting season, which is April through July. That happens to overlap with a large portion of our spring and summer, but anything early or particularly late that is ephemeral may not be represented in our study. And lastly, at the moment, we're primarily using presence and absence. We have this little estimate of how common things are found, but because we go through this step of amplifying the DNA, it's really hard to know exactly how common things were. And then on top of that, how common the plants were is not always proportional to the frequency of the DNA in their nests for so many reasons of layered steps between the plant being found in the wild and it ending up being sequenced by me 110 years later in the lab. So again, snapshots, but more evidence than we had before we started this study. We are trying to get into whether we can at least identify the frequency of plants per nest and help communicate this information to people who are doing restoration. And that's part of why I was so excited to be invited here, because I know many of you either help out with restorations, may work in restoration, or at least know somebody doing restoration, particularly around the margins of the San Francisco Bay. And I hope that with this information, we can start to bring back some transitional habitats like all those gold areas we saw around the South Bay, the North Bay, and the East Bay, not just for the plants, but hopefully for the whole ecosystem, including some of those cute animals that use it as refugia. I need to thank a couple people. Of course, Alex, my co-author and friend and colleague on this. Eliza Solomon at Santa Clara University did amazing work in the laboratory, and this wouldn't have happened without her. Ben Carter and Tracy Masewicz from San Jose State were the co-advisors of Alex's master's thesis and provided a lot of important resources for us, particularly their brains. Miranda Mellon is the person who originally put us in touch. She was working for San Francisco Bay Bird Observatory while getting her master's degree at San Jose State and meeting Alex, and she put Alex and I in touch. And Katie La Barbera is the ornithologist, scientist in residence at SFBBO. The Mid-Peninsula Open Space generously funded portions of this research. San Francisco Bay Bird Observatory paid Alex during that time. San Jose State gave him his master's degree, and Santa Clara has provided a lot of support, both logistical and technical. The Northern California botanists gave us a little grant, and the Stanford-based Bird Ecology Network, SOAR, is now continuing some of this research. With that, I hope some of you have questions because I'm really excited about this research, and thank you for the opportunity and hanging in there at this hour on a Thursday night.
Thanks, Justin. So let's see. Mary Ellen King wanted to know, does the new understanding of horizontal gene transfer confound your work?
Oh, excellent. Good, great question. Mary Ellen. So we now know that things don't always split the way my phylogenetic tree suggested, where one ancestor gives rise to two descendants, and so on and so forth until the tree of life is passed. Horizontal gene transfer is exceedingly common in bacteria. That's how antibiotic resistance often spreads on little round plasmids between different species. So instead of it being passed on from your parents, you actually can pick something up from the environment or an unrelated relative. It turns out it's pretty rare, to my knowledge, in plants. We do see it in parasitic plants. Parasitic plants have grabbed up DNA from the host which they parasitize. For example, a genus Aphelon, the broom rapes, they parasitize quite a few shrubs, and they have now been known to absorb portions of the DNA. If we had any plants that were known to participate in horizontal gene transfer, then yes, the DNA sequence would be misleading. But the chances that these angiosperms are picking them up seems really unlikely, given that our trees are very, they seem to reflect the natural history and the geography of those plants and not anything that would move them horizontally. Great question, and I think appropriate in some particular unique cases, but I can't think of any of the plants that we have, for example, that are parasitic, hemiparasitic. Maybe daughter, Cuscuta, if we ever bumped into it, but we didn't actually have any evidence of Cuscuta in any of our samples. Thanks for that thoughtful question. I'll think more on it if I come up with one. What else, Vivian?
Okay, this is a comment, but I'm going to follow it up. I'm going to add my own question to it. So this is from Carrie Olson, and she said, Justin, this is absolutely fascinating work, and you are absolutely spot on when you call this a method of time travel. It's brilliant. And so my question to go along with that, though, is so have you thought about expanding it to other habitats, other types of restoration work?
Yeah, it's a great question. And I'm in conversation with the Mid-Peninsula Open Space Regional District. It turns out Alex came up with this idea, which was like a perfect storm of a bird that only made a nest in this particular habitat. And we knew that we had old nests and this particular area needed to be restored. And so we're racking our brains now for analogous environmental circumstances, unfortunately, where we may not know anything about it. For example, a student asked me, "Well, can we use this for the fires in Los Angeles?" And I thought, well, let's think this through. Well, do we know what the plants were in that area? And we know pretty well what those plants were in that area. Unfortunately, these are where wildlands intersect with houses, so we don't need a nest from that area necessarily. But I think there are probably a couple of really unique habitats and unique birds, and I'm still thinking about it. So if anybody has any suggestions or ideas of a place, someone suggested dunes, because there's often a lot of human impact on dunes, and there are often a lot of birds that are unique to dunes, like take, for example, a plover. There's a lot of plovers that are very unique, and we might get some great insights into the dune habitat. That was one that a student offered, which I think would be worth pursuing. But the bird diversity is amazing. Everything from raptors to waterfowl to hummingbirds, right? Everybody makes a nest of different shapes, sizes, and content. And I think we could learn a lot. And as far as I know, we're the first ones to be sequencing the DNA from these bird nests. So there's a lot of opportunities. I just need some more some more ideas. If anybody has any, feel free to send them to me offline or raise your hand. Thanks, Vivian.
Vicky has her hand up, and I also have another question to read, but maybe if Vicky's might be relevant to what we were saying, so maybe let Vicky go first.
Well, um, well, first of all, plovers don't use plant material in their nests. They use stones.
Did you say hummingbirds or
No, plovers.
plovers? Right. Okay.
Plover,
There
yeah.
you go. Then I clearly, like I mentioned at the outset, this is out of my
But
room. We might have some, are there collectible nests of a dune bird that you would recommend?
I can't think of any right now. Maybe Verdon? I don't know. Just anyway. Oh, I might. Okay, so I I went over your paper that was referenced in the in this announcement, and you included Rosa Woodsii. Why did you do that?
Yeah, I think that has something to do with the blast results. And we don't think that the plant is actually Rosa woodsii. What ended up happening, to give you a little bit of the backstory, is that we were unsure what rose it was. And I was just going to have to leave it as Rosa spa, meaning we don't know which species. And that's when Alex took his results to Ben and Tracy at San Jose State. And Ben said, "Well, Alex, why don't you just go find some roses that we know could be the candidates because they weren't in GenBank, and Justin will help you get the DNA out and make them your references." And so he went out and did some plant collecting, made his herbarium specimens, and introduced them to the herbarium, and then we sequenced the DNA. And once we got Rosa californica in there, it matched perfectly. And then we overlapped it with the geography, those Calflora sites and now iNaturalist. And it's like a 200 to 1 chance that it's Rosa californica over
Yeah.
Nootkana
Yeah.
or Woodsii or anything else. Does that answer your question, Vicky?
Yeah, it's just that I thought Woodsii was so far away.
Yeah, I agree. That's the wrong thing. Agreed.
That's a long way for a song sparrow to go.
Good call. Thank you.
And, gosh, I really enjoyed your paper. Um, I think you just really think out of the box, and it's like really cool. Um, yeah, it's just cool.
Cool.
Thank you.
I'm glad that you liked it. And I should mention, I forgot to put the stuff on the slide. Maybe I'll send it through Vivian. We have another paper in the works, which isn't ready yet, describing the new data with the 72 species. But UC Santa Cruz graduate student in journalism, particularly nature journalism, decided that this was equally interesting and did her senior capstone design project on it and ended up being written up in Bay Nature, and she did a wonderful job. And then she made a really, really cool video out of it. And if there's a way I can share those with you, Vivian, and you can blast them out to your crew, if anybody's interested in getting a little bit more
Yeah.
details, something, those are some really nice entry levels, sort of gateways to this topic.
Yeah,
So your
Justin, please
so
send them
your
over and I'll post them. Yeah.
so your paper that's going to be coming out, will that be in the PLOS one also?
Yeah, it's still being written as we speak. I mean, it's literally in progress, and we haven't picked a journal yet. We'll probably send it to molecular ecology first, and if I remember, I'll pass it through the CNPS people when it does arrive and let you know.
And I have a question from Carrie, who is from the Santa Cruz chapter, and wanting to know who the USC student's name is. UCSC
Oh, excellent.
student's name is.
Well, let me stop sharing my screen here real quick. Hang on. And I can probably just put it in the chat. If you guys be patient for 1 second, I can find that. Oh, you guys have computers too. It's the journal is the article is in Bay Nature and it's called Bird Nest Detectives. And is it, do I have access to the chat, Vivian?
Yeah, you should. Just if you, there should be a chat if you go down to the bottom of your window. There's a
Yep, here
little
you go.
chat button
And
that
I'll
will.
send it to everyone. Her name is Brittany Miller, and Brittany was a graduate student in the journalism department there, and she got her degree and now writes for a newspaper out of state, I'm pretty sure. Oh, I guess I can share again if you want to see what it looks like. She did a wonderful job. She took a ton of time. There's Alex and his bird nest, and she really put the pieces together. And did a lot of on the ground work. We spent a lot of time out in the field with her at Alviso, for example, walking around, introducing her to the plants, and she went to the museums to see all the projects. So have a look at that, and I think you'll find the link to the video in there as well.
Thanks, Justin. Okay, you have another question. This is from Stu Woodward. And he says, I really like the implications of this excellent work. When you think about a wetland, it seems like DNA from plant tissue could come from very far away. So something like redwood would have traveled down waterways and come from much further up in the watershed. Have you looked at flora upriver from the areas?
Yeah, excellent points, Stu. And what you're hinting at is what we're finding in this field of eDNA or environmental DNA, that you can learn a lot from just DNA that's floating often in water or in soil or on the surface of other organisms, whether they be plants or animals. And I'd like to point out that no, the answer is no, we have not looked upstream, like going up the creeks. Um, but that's definitely where the redwood came from, or it could have come from pollen as they're wind pollinated. I think I also showed an oak and a pine, and although they could have been part of the nest, we didn't see any really distinctive things. I think the oak would be pretty unique if it was a leaf or associated with a flower or an acorn in the nest, and we would have been able to maybe note that it didn't look like a grass. And I'm pretty sure oaks, pines, and redwoods share one thing in common, which is they're wind pollinated, and therefore there's a lot of DNA moving through the atmosphere and then settling on water and then being blown probably by surface winds and high tides to this transitional area where, you can imagine this little like sort of line of all the floaties that have particles that have been dropped at the high tide line, and eventually that might have landed on the nest. So I was initially disappointed in finding things that I thought were probably not the transitional habitat and probably not actually ever growing in that area or used by a bird in the nest. And then I realized we still have oak, redwood, and pine DNA from like 110 years ago. Like I'm sure there's a glimpse of the past in there. And there's no doubt that those trees were there, but um, that part takes me back, and I appreciate that. Thanks, Stu, for the point about the upstream.
Thanks, Justin. Okay, another question, and I think you've already partially answered this one, but I'm going to go ahead and read it. It's from Jennifer Lamb, and she wants to know, where are you going next with this work? What do you hope to accomplish? And at what point in your research will you find that your research goals will be completed?
Yeah, I struggle with starting new projects where we're gonna, for example, maybe go look to a new habitat where we don't know what plants that grow there and the things we were talking about before. But clearly, that's on the margins of my expertise, as I think I suggested to use the nest of a bird that makes its nest out of rocks. So we are doing several other things. I would like to be more involved in the actual restoration practices around the transitional habitat so that my students, they help a lot with this research. And then they're always asking me, so now when do we do the work? And I said, well, you just worked so hard for several years on this project, but they want to see the transitional habitat get planted with these plants. They want to see it happening. So obviously we want to make sure we first do no harm and that we vet these plants really carefully, that they are most likely candidates. But I think I'd like to get more involved in that part, in the restorations and the monitoring of the restorations. And in fact, students were pushing me and asking me, well, you're not just going to monitor the plants, right? You're going to look for that mouse. You're going to look for the ridge-raised rail. You're in fact going to look for these particular birds to be using your plants to prove your point. I said, yeah, let's go. So that's one thing. But if you're curious, we have a couple collaborations with the San Francisco Bay Bird Observatory, and we're doing other sort of, I'll call it bird adjacent work because I'm learning a lot about birds. We'll just say that. There's a restoration in Milpitas around the Coyote Creek Field Station, and that's where Coyote Creek enters into the bay in the South Bay. and they've been doing this restoration for several decades, and they wanted to know if the birds were using the restoration. And as they capture birds in mist nets, and they ban them every weekend, they've been doing it for decades. When you put the bird into this very soft bag and then you tuck it in your jacket pocket to keep the bird warm while you take it back to the station and it gets its little anklet, the bird always poops. And it turns out the bird's probably trying to tell us what it's been eating. And we could use that information to determine whether it's been using the restoration or whether it's just been eating weeds. And that would help us understand sort of how the trophic levels are being restored, which plants are being used by birds more often. And sometimes it's obvious. They catch these birds, and I've been out there, and this like house sparrow, it just has its face is bright purple. It's just been gorging itself on sambucus, on elderberries. And other times it's more subtle. So we're actually having quite a bit of success sequencing the scat of birds collected at the restoration site and identifying it's about 50-50. About half of the DNA fragments in the scat are native plants and half are non-native, which is actually a really high proportion of natives given those habitats around the edges of the San Francisco Bay, which we know, unfortunately, are rarely 50% native. So we're doing a lot of that and some other fun projects. I work on flower color when we're not thinking about birds. So the Native Plant Society people are always my go-to when I need what did I need? I needed cardinal flower. I need some hummingbird flowers, and people were very generous in your list. Serve, you have sort of an email. I was looking for firecracker flower last
Oh,
spring,
yeah.
and I had a
Gardening with Natives group, right?
yeah, I had a deluge of places I could go find it, and it was so late in the season. So I'm so thankful that there's organizations and people like you guys out there, because yeah, you're doing amazing things. Listening to Vivian's list of activities, a Durka walk. Like, how many chapters claim to have a Durka walk? Now that's cool.
We love our Durka in this chapter. We really, really do.
Doesn't it give you hope in this time of year, especially when you see those flowers come out on the otherwise bare branches?
Oh, definitely. Okay, so Michelle Tillett has a comment and a suggestion, actually. She says, this presentation was wonderful. What a fun concept. I've read something similar about wood rat middens, and an idea perhaps that you for your next study. So,
Yes.
um,
Yeah, I love these ideas of going back in time and these wood rat middens and other places where stuff is preserved, particularly, yeah, like in caves, where you might be able to find DNA are absolutely fascinating to me, including like every once in a while Neanderthal DNA. We now know that well, many humans have about 1% of Neanderthal from the DNA found in some bones in Germany, and we know that they had unique alleles. One of them seemed to give pale skin and reddish hair to some of those Neanderthals. So yeah, I'm a big fan of time travel.
okay, I have a question for you from YouTube. This is from Ori Camp Carmi. I probably slaughtered the pronunciation of that name. I apologize. But here's a question. Maybe you can explore nests from chaparral. If I'm not mistaken, chaparral communities can really differ in composition, and wren tits do not stray far from chaparral.
Awesome. Hey, great tip. And especially a chaparral area that might have been maybe impacted where we're not sure what belongs there, which unfortunately I think there's probably many of those here in California because people like to live in areas that are often previously occupied by chaparral. I'll keep that in mind. Wren tits and
Yes, they
chaparral.
do have they stay very close within a small area.
Perfect.
Um, Okay, from back to Zoom, Sharon Beals says, I photo nests in all of the collections that you've worked in, except Harvard, including some collected by Grinnell. I've always wanted to learn what plants were used in their construction. Now I know it would be a dream. So I think she's very appreciative of your work.
Awesome.
And from Carrie, she says, that's the best part of the work, to see the plants in use by the other inhabitants of the habitat. I'm filled with hope.
Cool. I'm glad. We need a little.
And that actually, I think, brings us to the end of all the questions and comments. if I missed anybody on Zoom, if you can you can unmute and speak. But otherwise, I know we're also getting right to 8:30, so it is making probably maybe for a little bit of a late evening for some folks. Um, so maybe I'll just say thank you so much for doing the talk for us. I really appreciate it. It is really just such a fascinating way of looking at uh, a space and through time. It's just amazing that you've been able to do this detective work and find out more about the plants that were here before we muddled everything up. And it's pretty exciting that perhaps those plants will be back where they used to be as a result of this. So, oh, we've got another comment. The River Otter Ecology Project has done a genetic analysis of the scat collected from various study sites around Marin County and Point Reyes National Seashore. So that's just a comment about another place to get some genetic analysis has been done.
Yeah, thanks,
And
Marianne. Hey, and I want to invite your people, which I'm so thankful that your organization exists, and super appreciative to Vivian and her colleagues for inviting me. But if anybody has any suggestions or tips, I'm easily found on the internet, unfortunately. So please, you can find me at Santa Clara University in the biology department. Do send me an email or give me a call. I'd love to talk more about it.
okay, well, thanks everybody for joining us tonight. And thank you again, Justin, for this fabulous talk. All
All
right,
right.
everybody, I'm going to sign us off now. So good night.